Hello, my name is Jade Kast and I'm part of the Partha Lab. Our presentation is about a mutant microbial species that alters interspecies interactions in the zebrafish gut. The gut microbiome plays an important role in host health and development, with many diseases like diabetes and Alzheimer's disease being correlated to a disruption in the gut microbiome. Therefore, it is important to study the microbiome and understand the interactions within the microbial community that affect its structure and composition. However, interspecies interactions are difficult to study in vivo due to the microbiome's complexity and variability. One method to study interactions is by using germ-free model organisms, and then introducing a chosen set of microbial species to form a specific gut community. Previous work from my research lab used this method in the larval zebrafish gut, and found that interactions between commensal bacterial species pairs dampened in a multi-species community. One pair they studied was with the species Aeromonas, abbreviated as AE, and Enterobacter, abbreviated as EN. And they found a strong negative interaction where the presence of Aeromonas decreases the abundance of Enterobacter. In my work, I use a mutant Aeromonas strain referred to as AEMB for mucus blind because it doesn't recognize the glucanac sugar in gut mucus. We have seen that this mutant has a different spatial structure in the gut where it aggregates less than the wild type, so I studied how this characteristic would affect its interspecies interactions. I studied the interaction between Aeromonas mucus blind and Enterobacter as a pair and in a 5-species community to see how the interactions differ in these situations. To study these interactions, I used germ-free larval zebrafish, then inoculate a set of 2 or 5 bacterial species into the fish's flask water at 5 days post-fertilization. Then at 7 days post-fertilization, I dissect the gut out, which is shown on the left of figure 1. I plate the gut contents on agar plates so I can count how many colony-forming units, or CFUs, grow. As you can see in this plate image on the right of figure 1, each species is a different color, so I can count the number of colonies and then infer species interactions based on the abundance differences. Moving on to my results, figure 2a shows the log abundance of Enterobacter in green and Aeromonas in purple, in 1 species experiments, meaning they were the only species in the gut, and in 2 species experiments, meaning when both were present in the gut. The average Enterobacter log abundance shown by the black square drops by an order of magnitude when present with AE, which is consistent with previous work showing they have a negative interaction. Figure 2 shows the log abundance of EN in green and Aeromonas mucus blind in red in the 1 species and 2 species experiments. In this case, the average Enterobacter log abundance drops by 2 orders of magnitude. I also did experiments with the 5-species community to see how the interaction between Aeromonas mucus blind and Enterobacter would change, which are shown in figure 3. Figure 3a shows the abundance of all the species when Aeromonas is included, and figure 3b shows the abundance when Aeromonas mucus blind is included. When Aeromonas mucus blind is included, the average Enterobacter log abundance is lower, and there are more cases where Enterobacter was not present than when the wild-type Aeromonas is included. From these results, I concluded that the mutant Aeromonas mucus blind has a stronger negative interaction with Enterobacter than wild-type Aeromonas does. I saw this strong interaction persist in a 5-species community, which is in contrast to the previous work that showed interspecies interactions between species pairs were weaker in multi-species communities. This indicates the spatial structure of the bacterial species in the gut can influence interspecies interactions. However, the mechanism of this interaction is still unknown, so this is an area for future research that has already begun in my lab. The reason the interaction is different in the multi-species community and what mechanisms or factors play a role in it is also unknown and needs to be studied further. Ultimately, if interspecies interactions can be modeled, it may be possible to construct and select communities with traits associated with a healthy microbiome that could then be used in disease treatment. Thank you for listening to my presentation.